Hello friends, in this video we are going to discuss about gibberellin phytohormone. In this we will discuss about discovery of gibberellin, then chemical nature and structure of gibberellin, then physiological effects of gibberellin. We will also discuss about distribution of gibberellin in plants, its transport in plants. Then we will discuss about Gibberellin Biosynthesis pathway. At last we will discuss about gibberellin signaling pathway. Under this we will discuss about key components of gibberellin signaling pathway. Then signaling in two situations. First in the absence of gibberellin and second in the presence of gibberellin. So stay tuned. Gibberellin plant hormone. First of all, discovery of gibberellin. Gibberellins were discovered by Kurosawa in Japan on fungal disease of rice. He was investigating foolish seedling disease or Becana disease in the rice, which is caused by fungus Gibberella fusikuroi. Kurosawa was studying plant pathology. He saw a disease in rice plant which resulted in elongation of rice stems and leaves, and these were much longer compared to the normal ones. This was called foolish seedling disease, or in plant pathology Becana disease, and it was caused due to the fungal infection or due to the fungus known as Gibberella fusikuroi. So how Gibberella fusikuroi caused this foolish seedling disease? Through his studies, what he showed that it was due to a compound, and this compound is produced by the fungus Gibberella fusikuroi. This compound was named as gibberellin. Now the chemical nature of gibberellin, all are a group of tetracyclic diterpenoid compound. These are derived via Ant-gibberellin skeleton, which is the basic structure. Gibberellins are named as GA1, GA2, GA3 and so on in the order of their discovery. Gibberellic acid GA3. These are the first gibberellin that was structurally characterized. Currently, 136 gibberellins are identified from plants, fungi and bacteria. Structure. These have either 19 or 20 carbon units, and these are grouped into 4 or 5 ring systems. So these have two classes on the basis of the presence of 19 or 20 carbon atoms. Gibberellins having the 19 carbon atoms are the bioactive forms, and these bioactive forms of gibberellins differ in number and position of hydroxyl group, and sometimes CH3 and carboxylic group at different carbon atom of gibbane ring. Some of the bioactive forms of gibberellins GA1, GA3, GA4 and GA7. These have hydroxyl group on carbon number 3, carboxyl group on carbon number 6 and lactone bridges form between carbon number 4 and carbon number 10. GA5 and GA6, which are also bioactive form, do not have a hydroxyl group on carbon number 3. Most common bioactive form of gibberellin is GA1. So now the structure of the gibberellin. This is the basic structure of gibberellin, which is also known as the gibbane ring or Ant-gibberellin skeleton. Here there are 20 carbon atoms, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 and 20. So this is the structure of GA3. Hydroxyl group is present on carbon number 3, carboxyl group is present on carbon number 6 and lactone bridge is formed between carbon number 4 and carbon number 10. This is structure of gibberellin 5. All are same except that here hydroxyl group is not present on carbon number 3. Now the physiological effects of gibberellin. First is seed germination. Gibberellin causes the seed germination in dark. It overcomes light requirement by promoting the synthesis of variety of hydrolytic enzymes which solubilize the endosperm reserve. So there are certain seeds which are light sensitive. For them, light is required for the germination of the seeds. But if they are present in the dark, then this germination is inhibited. On the application of gibberellin, these seeds even germinate in the dark, because in the presence of gibberellin, the requirement of light is overcome. Now, this gibberellin promotes the synthesis of hydrolytic enzymes, which then solubilize the endosperm reserve, which is then used in the seed germination, and in this way seeds germinate. Rooting. At higher concentration, gibberellins inhibit root initiation in stem cuttings. Leaf expansion. Gibberellin causes leaf expansion and elongation. Fourth is internodal elongation. Gibberellin induces internodal elongation in genetically dwarf and rosette plant species. These are the nodes of the stem and these are the internode. Sometimes in the genetically dwarf plant, some genes are missing that produces gibberellin, or the concentration of certain natural inhibitor is high. So they are genetically dwarf. Because in the absence of these genes or in the presence of the natural inhibitor, there is deficiency of endogenous gibberellins. But when gibberellins are used or applied externally, then this endogenous deficiency of gibberellin is overcome, and this results in the internodal elongation. Since gibberellin is required in the internodal elongation. Fifth is bolting and flowering. Causes bolting and flowering in some long day plants in non-inductive condition, that is in short days. In the long day plants, for the bolting and flowering, long days are required. But when these are grown in the non-inductive condition, that is short days, then the bolting and flowering does not take place. If in this condition gibberellin is applied, then gibberellin causes the elongation of the stems. This then is converted into floral axis, which bears the flower primordia. This is known as the bolting. Similarly, it also results in the early flowering in long day plants. So this gibberellin overcome the requirement of long days in the long day plant. That is, it results bolting and flowering in the long day plants even in the short day conditions. Sixth is parthenocarpy. Gibberellin induced parthenocarpy. What is parthenocarpy? Parthenocarpy is development of fruit without the fertilization of ovule. Gibberellin has more effect on parthenocarpy, and it results in the parthenocarpy of those plants also in which auxine fails. Seedless and fleshy tomatoes and large size grapes on commercial scales are produced by gibberellin treatment. Seventh is dormancy of buds. In some cases, dormancy of bud is broken by gibberellin treatment. It is seen that in some cases, buds remains dormant during the unfavorable condition, and these breaks their dormancy only during the favorable condition. When gibberellin is applied, or when these buds are treated with gibberellin, then these break their dormancy even during the unfavorable condition. Light inhibited stem growth. Treatment of light grown plants with gibberellin stimulates stem growth. In most of the cases, it is seen that light grown stems are shorter, thicker and green, and dark grown stems are longer, thinner and pale. So what does this mean? That light inhibits the stem growth, because light lowers the level of endogenous gibberellins, and because the growth of the stem requires gibberellins, hence their growth is inhibited. But when gibberellin is applied exogenously, or when the light grown stems are treated with gibberellin, then it stimulates the stem growth. Fruit setting and fruit drop. Gibberellin increases the fruit setting and reduces fruit drop. Pollen germination. Gibberellin causes pollen germination. Also, it increases the rate of pollen tube growth. Biosynthesis of gibberellin. We will discuss about how gibberellin hormone is synthesized in plants. First of all, let us start with discussing distribution in plants. In higher plants, it is found, or it is present in all parts like shoots, roots, leaves, flower petals, anthers and seeds. Higher concentration of gibberellins are present in reproductive part, and in vegetative part, comparatively lower concentration of gibberellin is present. Immature seeds are especially rich in gibberellin. So it is the most important source for the isolation of gibberellins, or it is the most important part for the isolation of gibberellin. So the occurrence, it occurs in two forms, either in the form of free gibberellins or in the form of bound gibberellins. In bound gibberellins, it is usually present in the form of gibberellin glycosides. Now the synthesis and transport of gibberellins. Gibberellins are synthesized in young tissues of shoot and also in developing seeds. It is translocated by phloem and xylem. Movement. We have seen in auxins that these show polar movement, but gibberellins do not exhibit polarity. These can move both up and down the stem, and these are translocated in bound form, not in the free form, mostly in the form of gibberellin glycosides. Now let us see gibberellin biosynthesis pathway. Gibberellin biosynthesis pathway involves various steps, and the different steps of the pathway takes place in plastid, followed by endoplasmic reticulum, followed by cytosol. Now let us see this pathway step by step. First of all, trans-geranyl diphosphate is converted into N-copolyl diphosphate. This step is catalyzed by the enzyme N-copolyl diphosphatase. Then N-copolyl diphosphate is converted into N-corine, catalyzed by the enzyme N-corine synthase. These steps takes place in the plastid in higher plants. N-corine is then converted into N-corinoic acid, catalyzed by enzyme N-corine oxidase. N-corinoic acid is then converted into N7-alpha hydroxycorinoic acid. This step is catalyzed by the enzyme N-corine acid oxidase. Then this N7-alpha hydroxycorinoic acid is converted into Ga12-aldehyde, catalyzed by the enzyme N-corine acid oxidase. Here what we see that Ga12-aldehyde is converted into Ga12, which is a non-13 hydroxylated gibberellic acid. So here Ga12 is present at the branch point. From Ga12, one pathway goes this way, and from Ga12, due to the hydroxylation on carbon number 13, we get Ga53, which gives another pathway which goes this way. These steps takes place in the endoplasmic reticulum. So here GA12 and GA53 are precursors for the two pathways. GA12 is the precursor of non-13 hydroxylation pathway and GA53 is the precursor of 13 hydroxylation pathway. So if we see this pathway, non-13 hydroxylation pathway, GA12 is converted into GA15, which is then converted into GA24, and GA24 is then converted into GA9. All the steps are catalyzed by GA20 oxidase enzyme. GA12 is oxidized on carbon number 20 and we get GA9. If we see this pathway, 13 hydroxylation pathway, GA53 is converted into GA44, which is then converted into GA19. Then what we get? GA20. All these steps are catalyzed by GA20 oxidase enzyme, and here also GA53 is oxidized at carbon number 20. So now what we get? GA9 from the non-13 hydroxylation pathway and GA20 from 13 hydroxylation pathway. So in the final step, GA9 is converted into GA4 and GA7, catalyzed by the enzyme GA3 oxidase. In this, GA4 is more important, that is gibberellic acid 4. So if we see this pathway, here GA20 is converted into the final product GA1, gibberellic acid 1, which is an important bioactive gibberellin. GA20 is also present at the branch point. Here we get GA1. Also GA20 is converted into GA5, catalyzed by the enzyme GA3 oxidase. Then GA5 is converted into GA3 and GA6. These steps are also catalyzed by the enzyme GA3 oxidase. So these two pathways, non-13 hydroxylation pathway in which GA12 is the precursor and 13 hydroxylation pathway in which GA53 is the precursor. These two pathways takes place in cytosol. These are the enzymes and their full form that are involved in the biosynthesis of gibberellin. Now let us understand biosynthesis of gibberellin once again. Different steps in this biosynthesis pathway takes place in plastid, endoplasmic reticulum and cytosol. It starts with trans-geranyl diphosphate, which is then converted into N-copolyl diphosphate, and this is then converted into N-corine. These steps takes place in plastid. Then this N-corine is converted into N-corinoic acid, which is then converted into N7-alpha hydroxycorinoic acid, which in turn is converted into GA12 aldehyde. Then this GA12 aldehyde is converted into GA12. This GA12 is present at the branch point. From this GA12 we get one pathway, or this GA12 is converted into GA53 by the hydroxylation of carbon number 13. So what we get? GA12 and GA53. These all steps takes place in endoplasmic reticulum. GA12 is the precursor of one pathway known as non-13 hydroxylation pathway, and GA53 is the precursor of the another pathway known as 13 hydroxylation pathway. And these two pathways takes place in cytosol. So in this non-13 hydroxylation pathway, GA12 is converted into GA15, which is then converted into GA24, and then GA24 is converted into GA9. This GA53 in 13 hydroxylation pathway is converted into GA44, which in turn is converted into GA19, and then GA19 is converted into GA20. So from one pathway we get GA9 and from the other pathway we get GA20. and both are obtained by the oxidation of the carbon number 20. Then this GA9 gives the final product. It is converted into GA4 and GA7. Among these, GA4 is more common. Here GA20, it is present at the branch point. From this we get GA1. Also this GA20 is converted into GA5 by the oxidation of carbon number 3. This GA5 is then converted into GA3 and GA6, and among these GA1 is more common. In this way, biosynthesis of gibberellin takes place. These are the enzymes and their full forms which are involved in different steps of biosynthesis of gibberellin. Now let us see gibberellin signaling pathway. We have already discussed that in plants, gibberellin regulates seed germination, leaf expansion, stem elongation, flower induction and seed development. So now the question comes that how gibberellin regulate these physiological functions. It is brought about by gibberellin signaling pathway. Like all signaling pathways, gibberellin signaling pathway also has certain key components. Let us discuss them one by one. First is ligand. It is signaling molecule. Here it is gibberellin. Second is receptor, which binds with ligand. Example GID1. It was first identified in rice and the full form is GA insensitive dwarf 1. It is soluble receptor. In Arabidopsis, there are 3 orthologs of GID1 at GID1A, B and C. GID1 have high affinity for bioactive gibberellins. These bioactive gibberellins we have already discussed. You can refer my video. Gibberellin binds to a specific binding pocket on GID1. Now third is transcription factor. It helps in transcribing necessary genes. One of the example of transcription factor is PIF, phytochrome interacting factors. These negatively regulate light signaling and are strong promoters of elongation growth. Fourth is repressor. It suppresses the ability of transcription factor to enhance gene transcription. Hence, these are repressor of plant development. That is, these repress seed germination, seed growth, flowering, etc. Here the repressor is DELA protein. In rice it is SLR1 and in Arabidopsis it is GAI and RGA. And the effect of repressors are reversed by gibberellin. So we can conclude that DELA proteins are class of nuclear proteins that act as regulators of transcription and suppressors of gibberellin signaling. So how it functions? It functions by stopping binding of transcription factor to DNA or by promoting their degradation. So now let us discuss how signaling takes place. There are 2 situations. First situation, when gibberellin is absent. In the absence of the gibberellin, no gibberellin comes and binds with this receptor GID1. Hence, this DELA protein, which is inhibitor, binds to transcription factor and prefoldins. As a result of which, these are not able to transcribe genes. So, signaling is off. Let us understand this with the help of figure. Gibberellin is absent. No gibberellin binds with receptor. Hence, this DELA protein binds with the transcription factor. This transcription factor is not able to transcribe genes and hence we say signaling is off. Now the second situation, when gibberellin is present. When gibberellin is present, it binds with GID1, that is receptor, which in turn binds with DELA protein. So now this DELA protein does not bind with transcription factor and prefoldins. They are free, they are able to transcribe genes and hence we say signaling is on. let us understand this mechanism in detail through this flowchart. Gibberellin binds with GID1, that is receptor. This binding results in the interaction between GID1 and DELA protein. Hence, it forms GA-GID1 DELA complex. And this complex formation results in changes in structure of DELA protein. Now, DELA protein is able to bind to F-box protein. This F-box protein catalyzes addition of ubiquitin to DELA or ubiquitination of DELA. This ubiquitination of DELA enables degradation of DELA protein via 26S proteasome. Due to the degradation of the DELA protein, transcription factor is free or it is released. Now, this transcription factor is able to transcribe genes, or we can say that signaling is on. Let us understand this with the help of figure. Gibberellin binds with GID1, which then binds with DELA protein. So, GID1 DELA complex is formed, which then enables DELA to bind with F-box protein. Here it is SCF1. This F-box protein catalyzes ubiquitination of DELA. Due to the ubiquitination, now DELA is degraded by 26S proteasome. So, it is degraded. As a result of which, now the transcription factor is released or it is free. Now, it is able to transcribe genes and hence transcription or signaling is on. Now, let us understand binding of gibberellin to receptor and how gibberellin GID1 DELA complex is formed. C3-hydroxyl on gibberellin makes contact with tyrosine 31 in GID1 binding pocket, and this causes changes in GID1 structure. This change in GID1 structure causes a lid on the GID1 to cover gibberellin binding pocket, and when gibberellin binding pocket is covered, it results in the exposure of surface, which enables the binding of GID1 to DELA proteins, and in this way gibberellin GID1 DELA complex is formed. So, this is all for today's video. In the next video, we will discuss about cytokinin plant hormone. So, stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.